Hello, everyone. I'm Wang from Kim5O's lab. Here, I would like to share a story we published in Nature Structure and Molecular Biology in 2023. In this work, we conduct transcript-wide RNA m6A modification analysis in mouse oocytes and early embryos. More than 150 RNA modifications in living cells have been identified to date. Of them, m6A is the most abundant modification in the internal region of messenger RNA. m6A is involved in many gene regulatory processes such as RNA splicing, transport, degradation, and translation. Understanding the gene regulation during mammalian early embryonic development has been a biomedically important research goal in the field. Since the first transcript data was released in 2013, other types of omics data, including DNA methylation, chromatin excitability, histone modifications, and 3D genome structure, have been reported over the years. However, before we initiated our project, there was a knowledge gap of transcript-wide m6A profiles in mammalian early embryos. Even though our m6A specific immunofluorescence staining experiment told us m6A does exist in mouse oocytes and early embryos. This is because the genetic materials, that is, cells or RNAs, are limited in this scenario, and there is no sensitive in vivo m6A mapping method to allow transcript-wide m6A mapping using small amounts of cells or RNAs. To solve this long-standing technical challenge, our collaborators at the University of Oslo developed a picogram-scale m6A mapping method called pico-me-RIPseq. Pico-me-RIPseq enables transcript-wide m6A mapping with as little as 100 picogram polyase-selected RNAs or small amounts of mammalian cells. In the first step of this method, cell lysis and the depletion of genomic DNA and ribosome RNA are conducted in the same tube to reduce the loss of the materials. Next, full-length RNAs are shared into small fragments with the optimized sonication condition. After RNA fragmentation, we take about 10% of RNA materials for regular RNA sequencing. This will serve as input control for downstream data analysis, such as m6A peak calling. The remaining 90% of RNA materials are subject to immunoprecipitation, that is IP, with the optimized antibody specifically against m6A, followed by careful washes to remove background noise. Furthermore, one-tube sequencing library construction is performed for input and IP sample respectively. After Illumina sequencing are performed bioinformatics and data analysis, including quality control, alignment, peak calling, and other downstream analysis. Using pico-me-RIPseq, we generate transcript-wide m6A profiles in mouse oocytes at GV and M2 stages, and early embryos at zygote, 2-cell, 8-cell, and blastocyst stages, with 2 biological replicates for each stage. For example, as clearly shown here, the consistently expressed and m6A modified at all stages, the gene CTCF, which is an important transcriptional repressor and chromatin regulator. Another example is DNMT3A, which is a DNA methyltransferase. We also see the beautiful m6A profiles located at the stop codon for gene USP2 before 2-cell stage, and also for the gene KLF4 from 2-cell to blastocyst stages. Pearson correlation analysis tells us the consistency between 2 biological replicates for both input sample and IP samples is pretty high, as an example shown here at the GB stage. This highlights the robustness of our method and the data we generate in this study. Moreover, based on the transcriptome-wide m6A profiles, we can see the distinct clustering of different stages as shown in this principal component analysis. After looking at the overall m6A profiles and the evaluation of data quality, we performed the identification of m6A peaks using IP sample versus the corresponding input control sample. For each stage, on average, we detect more than 10,000 of m6A peaks. Same to the previous observation in other cell types and tissues, the peaks we identified in all stages are highly enriched surrounding the stop codon of protein coding gene. Meanwhile, we found that the enriched motifs at all stages are the classical well-known m6A motif RRACH. In terms of m6A modified genes, we found that on average, around 5,000 genes are m6A marked in each stage, and there is a clear switch of m6A status from zygote to two-cell stage, which might be explained by the activation of the zygotic genome and the reprogramming of the transcription after fertilization. We further focus on those stage-specifically expressed genes and classify them into 8 groups based on their expression pattern across the 6 stages and analyzed their m6A status. Interestingly, we found the stage-specifically expressed genes with m6A modification are significantly involved in transcription-related processes, while those without m6A modification are enriched in basic metabolic processes. We further demonstrate that comparing to the other types of genes, stage-specifically expressed transcription factor coding genes are indeed more likely to be modified by m6A as shown in the red dot here. Furthermore, we look at the m6A modification on those very important transcription factors, which are critical for the first lineage specification event, that is the differentiation of bases into the trifactodome and the inner cell mass, and the second cell fate determination from inner cell mass to epiblast and primitive endoderm. We found that almost all its important regulators are m6A modified, such as CDX2, which is important for trifactodome, and gene NANOC, which is important for both first and second lineage specification. After fertilization, there are 2 important events during the mammalian oocyte and early embryonic development. The first is the degradation of maternal inherent transcripts, also called decay. The decay can be further grouped into 3 types. The first type is the maternal decay, for short here called MDK, where RNA are greatly degraded during the oocyte maturation. The second type is the zygotic decay, for short here called ZDK, where RNA are greatly degraded after fertilization. And the third one is continuous decay, for short here we call CDK, where the RNA is continuously degraded over oocyte and early embryonic development. The second event is activation of zygotic genome, that is ZGA, which occurred at around 2-cell stage, where the transcription of genes from both alleles are activated. We try to look at the m6A profiles on decay and ZGA genes. Based on the gene expression profiles from GV to 2-cell stages, using the same criteria as described in the study of the last slide, we identified 434 MDK genes, 1,722 ZDK genes, 137 CDK genes and 726 ZGE genes. We found around 50% of ZDK genes and the ZGE genes are m6A modified, while the ratio of m6A modified ones is a little bit lower than 50% for CDK gene, and is less than 30% for MDK. Here we show some examples. We can clearly see the m6A profiles at the specific stages. For example, MDK genes GLCE, ZDK genes TBC1D8, CDK gene GCNT4, and also the ZGA genes KLF9. In addition to m6A, one type of small RNAs with 22 nucleotide lengths in animals called microRNA that plays very important regulatory roles in post-transcription processes, including RNA stability and translation through base pairing with the sequence of 3 untranslated regions of messenger RNA. Years ago, one study from my PhD lab at Chinese Academy of Sciences showed that microRNA and m6A can work together to regulate the reprogramming of cell fate. We wonder if microRNA has a tendency to target m6A modified DK and ZGA genes. Our analysis results show for both DK and ZGA genes, m6A modified ones are more likely to be targeted by microRNA as compared to those unmodified ones. This preference is statistically significant as calculated using Fisher's exact test. Further, comparing to m6A modified ZGA genes, microRNA has significantly higher preference on m6A modified DK genes, which may indicate the potential co-regulatory roles of microRNA and m6A in degradation of maternally inherent RNAs. For the genes which are occupied by both m6A and microRNA, gene enrichment analysis shown the opposite functions for DK and ZGA ones, as highlighted by green and blue box. DK genes are involved in positive regulation of transcription, while it is negative for ZGA, and the DK genes are involved in negative regulation of cell proliferation, while it is positive for ZGA genes. In addition to the transcript from regular genes, we want to ask if other types of transcripts are also m6A modified, such as those from the actively transcribed RNAs from repetitive genomic regions in mouse oocytes and early embryos. Transposomal element, somebody called them as jungle DNA, which contribute 45% of human genome and 37% of mouse genome. Most of them are retrotransposons, including the long-term repeat, LTR, long interspread nuclear element line, short interspread nuclear element sign, and some evolutionarily young subfamilies from its retrotransposons, such as LAN1, ALU in human, and LTR can be transcribed into RNA and then reverse transcription into DNA and finally reinserted into the host genome through a copy-to-paste mechanism. In the mouse life cycle, there are two waves of the epigenetic reprogramming. One occurred during the germ cell development. Another occurred during early embryonic development. Coupled with the two waves of epigenetic reprogramming, some retrotransposons, such as LAN1, are transcriptionally activated when the DNA methylation goes down at some specific developmental stages. In 2021, 3 papers published in Nature show that in mouse embryonic stem cell, m6A can safeguard cell identity through targeting retrotransposon-derived RNAs. We want to ask if m6A can also target the retrotransposon-derived RNAs in the developmental stages we studied here. We first investigate the relative enrichment of m6A peaks on full-length copies of 3 types of retrotransposons. We found that before 8 cell stage, comparing to line and sign, m6A more prefer LTR. In 8 cell and blosted embryos, line shows relatively higher m6A enrichment. At the family level, we found that m6A is relatively enriched in ERVL at the 2 cell stage. The enrichment patterns of m6A observed for LTR and line over the development stages are mainly contributed by ERVL, MALR, and R1 families, respectively. In addition to calculate the m6A peak enrichment for each retrotransposon copy of locus, we also calculate m6A signal as a log2 value of normalized rate coverage from an IP sample versus input sample, giving a locus. If its m6A signal is positive value, we consider it as m6A modified, and further calculate the ratio of the loci with positive m6A signal for each subfamily. As the purple-colored heat map shown here, around 90% of loci for MTA subfamily have positive m6A signal value from GV to 2 cell stages. This similar ratio is also observed for the 2 cell specifically activated MERBL subfamily. In addition, the young R1 MDT subfamily also shown high m6A enrichment in blastocysts. Further, focusing on the locus level of MTA subfamily, found around 90% expressed full-length MTA loci are m6A modified, and near 90% of full-length MERBL are m6A modified at the 2 cell stage. When looking at the distribution of m6A along the full-length structure of MTA, we identify that the whole region of the MTA seems to be occupied by m6A. This is also can be supported by the very high frequency of multiple GGACU motifs along the MTA sequence. While for MERBL, m6A more prefer the middle 3' region, where GGACU motif is relatively frequent than other regions. In summary, in this study, we generate m6A landscape in mouse, oocytes, and early embryos. We show that m6A widely modified stage-specifically expressed transcription factors, especially for those important regulators for lineage specification. We found microRNA and m6A can co-target decay and ZGA genes. The mechanism behind this can be investigated in the future. In addition, retrotransposon-derived RNAs such as MTA and MERBL are also frequently modified by m6A. m6A might be a potential regulator for controlling the retrotransport activity in early embryos. Overall, together with the other 2 studies published in Nature Cell Biology and Nature Communications, our study opens a door for future functional studies of m6A-related gene regulation during mammalian early embryonic development. Finally, we would like to thank our collaborators from University of Oslo, Wang and Angel. Especially Angel developed the pico-MIRIPseq experiment method and generate sequencing data used in this study. We also thanks for the funding support of Ohio State University, University of Michigan, and the NIH, as well as the grant from the Norway government for our collaborators. Thanks.